Hello Bio 120. This week we are going to take on running a protein gel. Now a lot of the concepts in running a protein gel, the general concepts, are the same as what we did when we ran our DNA gel. Now we're going to use a slightly different matrix. In DNA gels we used agarose. In protein gels, we're going to use polyacrylamide gels. And that's what you see here. We bought these pre-made cast gels, and they come from Biorad. They're pretty expensive, so I'm actually not going to take this gel out of its packaging, but I'll try and show you how it's going to fit into our gel apparatus as best I can with it still in its wrapper here. But what you see in front of you at the top is a green comb, and we used combs with DNA gels. This comb is performing the same function. It's going to leave little indentations in our gel. When this gel was made, molten polyacrylamide solution was poured into the plate, and the comb was then inserted while the polyacrylamide was still melted. So when we pull it out later in this protocol, it will actually leave those little wells, those little indentations. And so that's what you see in the green comb. You also see a green seal on the bottom. And so this is going to be something that we remove when we actually go to run the gel. But when you hear me talk about the seal, I'll grab this gel again, but if I forget, that's what I'm referring to, is this little seal on the bottom. And that's necessary because we need, to make the gel, we need to seal the bottom of this plate casting system is actually open, and so in order to make the gel, we need to seal that up. But we need to then peel it away when we go to run the gel. And so I mentioned the comb, mentioned the seal. Well, the actual plate layout that our gel is in, this is a very thin gel, and you can kind of see that on its edge. It's a very thin gel, and what's happened, or the way this gel was made, is that it was made using a sandwich. A tall plate and a short plate were sandwiched together using some little spacers such that the tall plate could, the short plate could leave space then for the comb to fit in. And so within these two plates sandwiched together is a little space, and that's where our molten polyacrylamide was poured. And so we have now solidified polyacrylamide within this space. But it's a very thin, very fragile thing. And so what we're going to do, the reason I mention the tall plate and the short plate in detail, is that it'll become important for when we're actually putting this gel into our casting system, into our running system. And so this is a part of our running system. Our running system actually comes in three pieces, two of which are gel holders, and one of which I'll refer to as the cassette. And so just to get at how to load a gel into one of these gel loaders, you'll notice that we have some spacer screws here. We've got our clear plate in the middle, and then our vanilla colored runners on the outside. And so the way we are going to set up this gel is when we take it out of the packaging, we are going to peel off the seal on the bottom, and then we are going to load this with the clear plate. Make sure your screws are unscrewed so that the clear plate can move. And within these little spaces here is where we are going to put our plate. And we're going to put our plate in there such that the short plate faces away from the screws. Another way of saying that is that our comb is going to be facing away from our screws. And so you can kind of see the tall plate in blue or gray there. The comb is loaded above the short plate, and we're going to put this in to our gel holder such that the comb is on the opposite side of the tall plate from the screws. Alright? So once that's in there, once we have the seal off the bottom of the gel, the seal off the bottom, and our short plate is facing the inside, we're going to screw in. We can lay that down, and we'll actually screw in our clear plate so that it's snugly sitting against our gel holder. Alright. And so with that set, and you can just imagine that our gel is in here, we're going to take our cassette out. Now notice that the top of the gel and the comb end of the gel overlaps or is at the same end as these big nubs here. There's either end, there's short nubs at this end, there are taller nubs here. Those taller nubs are actually what are going to fit into our cassette, and then once those tall nubs are in, we can push down and snap our gel holder into our cassette. Okay, and that is how we'll put these two pieces together. And you'll notice that on our cassette, I'll just flip it over here for a second. On our cassette, we have a little wire running along the bottom here. And that's going to be one of our electrodes. And so this gel is actually going to run vertically, and we'll see that when I load it into the tub. We're going to set this upright, our well's at the top, and our gel is going to be run vertically. So that's another small difference between DNA gels and protein gels, is that protein gels tend to be run vertically. Okay, so we've got this other half of our cassette here, and we're not just going to leave that blank. Each of these cassettes has two of these plate holders, and the plate holders can also act as a spacer, and that's what we're going to use the other plate holder for. Each group, each table I should say, is going to use a single gel, and therefore each table is then going to need to use a spacer here. So again, with the taller nubs at the top of the cassette, you slide those in, and then pushing against the bench, just push those and snap the smaller nubs into place. Okay, and that forms our gel holder cassette apparatus. Alright, so with that, we are going to put that into a tub of buffer. So again, just like our DNA gel, we're going to submerge our protein gel in a running buffer. And that running buffer has ions in it, and it's going to conduct that electricity and pull our samples, pull our proteins down into our gel. Okay, so you'll notice I set the entire apparatus in one of these tubs down the middle, and these tubs have these little lips on them to kind of keep the whole apparatus in place. And so when it's all said and done, your setup should look something like this. Okay, so now with that, looking top down at this, we have an inner chamber and we have an outer chamber. And we are actually going to fill both of those with running buffer. First, we're going to start out by filling our inner chamber with running buffer, fill that all the way to the top, of these clear plates. And then you're going to fill the outer tub, fill the outer region. And so with that all filled, notice I didn't tell you to take the comb out yet. With all that filled, the comb should come out very easily at this point, because the liquid, the buffer, should help lubricate that comb a bit so that you can pull it straight out. But again, just like with DNA gels, you want to pull out that comb very carefully and straight up. Okay, these gels are very fragile and will tear. The wells will tear even more easily than they did with the DNA gels. So use two hands, I would suggest, and pull that comb straight out. So with the comb removed, you're going to expose the wells on your gel. And so looking at it side on, you're going to see something like this. You're going to see all these little indentations that represent the wells of this gel. And so the well containing part of the gel and a bit down into the gel itself, that's actually a slightly different substance, a slightly different, has a slightly different makeup than the rest of the gel, and it's actually referred to as the stacking gel. And I don't want to get into the purpose of the stacking gel, but know that when I refer to the stacking gel, that I'm referring to this top portion of the gel, and that'll become more of a more prominent part of the protocol next week. But the stacking gel is the top part that contains the wells. So into the stacking gel, you're actually going to use a special type of pipette tip. Notice how long this pipette tip is. And we're using our P20 here, and these special pipette tips that will be out in the lab. We are going to use those to load our samples, and so that's going to look something like this. It might be kind of difficult to see the white on white background, but we are going to insert our pipette tip down into the well. And again, just like DNA gels, you don't need to get the tip all the way to the bottom of the well to expel your sample. You can be, maybe just halfway down the well to expel your sample, and the sample buffer that your sample is in will cause your sample to flow to the bottom of the well, which is where we want it to end up. Now, when you are loading your sample, I highly suggest that you just drop a very little bit at first. Okay, just expel a very little bit of sample at first to make sure that it drops down into the well, because it's very easy to miss the well and be somewhere in this inner chamber and miss your well completely. So expel just a little bit of sample initially. Make sure that it then settles at the bottom of the well, and it'll form a little band. And then once you see that, then add the rest of your sample. Okay, and so you'll do this. Take turns with your partner. Every table, as I mentioned, will load their samples into a single gel, and I'll refer you to your lab manual as to how to do that. You'll also load a marker lane, so one of these lanes, one of these wells, will be dedicated to a protein marker. And once you've completed loading all of your samples, and you've done this and coordinated with the other groups at your lab bench, you'll have a loaded gel within a gel running apparatus submerged in running buffer. Okay, and so with that all set, it's time to add the top and add the leads to our system and connect this to a power source. And so with that, you'll notice that there are colors associated with these electrodes. And so when you put on the top here, you are going to load the top black to black, black to black, red to red. I know this is a little discolored. These are old boxes, but you'll definitely be able to tell which one is black. And so with that, you're just going to add the top, and you will be able to then take the leads over to your power source, and there should be one on your bench somewhere, and again load the leads red to red, black to black. Okay, and then I'll refer you to your lab manual for the settings that you then set up on your power source. Okay, so then once that's all set up, you should see bubbles coming from bubbles forming inside of your running apparatus, and that means your electric current is running through your system and your proteins are being pulled through your polyacrylamide gel. And that's how you run a protein gel.